Dank u wel. Ik ga verder in het Engels gaan, dus I will switch to English now, so that everybody will be able to follow at least what I'm going to talk about. So I'm very happy to see a lot of familiar faces, and today I'm actually going to talk about revolutionizing viral and bacterial diagnostics in veterinary medicine using nanopore sequencing. I'm not going to do all the work today. I will first start with giving a short overview. I will introduce the project a little bit, talk about what the objectives were in this work. Then I will talk about science. Then we have some conclusions, and then it's time for questions from the jury members. Now, as I said, I'm not going to do all the work today myself. I will also ask some questions to the audience. Who has done a quick SARS-CoV-2 self-test at home? Just raise your hand, that's what I just want to see. So that's what I expected, almost everybody has done this. Now, just for you to give you an idea what is happening in this little white block at home, we actually have an antibody that is at the bottom attached to it, and it allows us to bind to the specific virus or bacterium that we want to target. That is actually what happened against the SARS-CoV-2 in this small little test that we have. Now, the same approach can be done to test for the immunological response that we can get from a viral or a bacterial infection. And then we are actually not going to look at the presence of the virus or a bacterium, but we're actually going to look at the antibodies that are produced within our own body. Because if we get an infection, we actually get antibodies that will help us to protect us against the infection that will happen. Now, if we think about this type of diagnostics, because my talk is going to be about diagnostics, this is done in about 89% of all diagnostics. In veterinary medicine, more focused on cattle and on pigs. So we actually have a lot of these tests. You might wonder, why is this applied in so many cases? It's quite quick. We can actually have a low cost, so we don't have to pay a lot of money for it. They have a quite okay sensitivity specificity. One of the disadvantages is we cannot test for antibiotic resistance, because I will be talking about antibiotic resistance in the future as well. So we cannot test for this here. We can somehow quantify the amount that is in there. As you could see, sometimes you had a very bright red light on your line on your test. Sometimes it was just very faintly present, so that meant there was not that much virus present in your test. And then we are somehow able to detect vaccination as well. Now, a little remark here. If we want to detect the vaccination status of an animal, we actually need to make sure that we have paired sera here. This takes about 3 weeks. That means you require 3 weeks before you can actually determine if you have an ongoing infection or you're actually detecting the vaccine itself. Now, second question, who has done a SARS-CoV-2 PCR test and received the results of these PCR tests? That's what I expected, almost everybody. Well, the questions will get more difficult, don't worry, because my next question is about who has gotten the PCR results of the flu, influenza A PCR. I might see some of them because they started to test for them as well in the hospitals, but it's not that common. So how is this test actually working? Before I go into that, we really have to dig in a little bit into the essentials of life. Because every organism that is living, bacteria, viruses, human, anything, it is actually built up out of the exact same code. It is A, C, T, G. 4 codes, that's as simple as it is. Now, some RNA viruses and RNA in general actually has a special change in this T letter. It becomes a U, but they are very similar to each other. Now, all these codes, they can actually build up a specific virus or a specific bacterium. So a sequence of all these nucleotides that we call them as well, of this 4-letter code, it actually allows us to build a specific virus. So what do we get? A unique code, for example, the SARS-CoV-2 virus. If we think about the influenza virus, it also has its unique code built out of the exact same 4-letter code. If we think about bacteria, they have a way bigger genome, but they also use this exact same code. So we can actually use this to diagnose them. And this is exactly what we use in our molecular detection, which is in our case the PCR test. If we have the virus, of course, we're not going to look at these 30,000 nucleotides that are in the coronavirus. We're just going to look at the small segment, which is indicated in black here. So if we know which virus we actually target, we can go and look for that specific sequence because we all know how the sequence looks like of the SARS-CoV virus, so we can easily target it. Now, the problem is with PCR tests is that it remains a black box. We don't get any information on the sequence itself. We can just say if something is positive or if something is negative. There's very little people in the room that knew if they had an Omicron infection, for example, or if they had a Delta infection. You just knew, I got infected. I'm positive. I tested positive. So this information allows us to actually do the PCR test, which is very quick. It's quite expensive. It's more expensive than the technology that I discussed before. It's highly sensitive and specific. And we can do some antimicrobial resistant testing, but it's not that common for all bacteria that we can test it for. We can also quantify, because that's one of the powerful things of the PCR and the molecular methods, that we can really dig into the quantification of the presence of viruses of bacteria. And there's some tests available to detect vaccinations or vaccines itself, but they are usually not that good. Now, if we think about this quantity, let's assume there's two people infected, one with a low amount of virus, one of a high load of the virus. If we would represent this in the black boxes, we would get a person that is actually medium loaded and one that is having a high viral load of the SARS-CoV-2. If we would test the same person 7 days later, the first person might actually not have any infectious virus anymore. However, because we have this mucus in our nose, there might still be the genetic material present in the nose, so we actually might still detect it with the PCR test because we are targeting the specific genetic material of the virus itself, while person 2 will remain at a low load, but it's still infectious. There the question is, what's the cutoff to determine if somebody is still infectious or is non-infectious in this case? Now, you might think, what am I telling here? You will see and put it in perspective in the next slides because we're going to make a kind of comparison later on. My next question, who has ever been diagnosed with a mycoplasma pneumoniae infection? I see one hand raising, yes, one lucky person there. I'm not surprised. I'm not surprised at all, because actually this is something that is very rarely diagnosed because it's not that easy to diagnose. We could use the PCR to detect its presence and then we would know it is there, but what is more commonly used for bacteria is to culture them. We actually want to grow and see if they are really alive, if the bacteria are alive and potentially infecting the host again. Now, for some bacteria, this goes quite smoothly. It can go very quick. It can take only 24 hours before you have an infection that can be diagnosed. Now, if we think about this mycoplasma that I said, some of them, it takes up to 14 days before we get to the diagnostics. This is the case for the pig version of the mycoplasma. For others, we already have speeded up by doing a lot of research to diagnose them in a quicker way. However, it still is a very cumbersome task. We can do the same for viruses. It's actually not done that often to isolate the virus itself. It's interesting from a vaccine perspective, but actually it takes too long to imply it as a kind of diagnostic tool. Now, making the comparison again, as indicated, for some bacteria, it's quite time efficient. Within 24 hours, we can have an efficient diagnostic. The cost is somewhere in between the serology and the molecular tests. Sensitivity specificity is also somewhere in between. Antimicrobial resistant testing is definitely possible in this case because that's actually the power of this type of diagnostic test. Now, if I zoom in for a moment on this, we can just add antibiotics to the medium to see if they can grow in the presence or if they can grow in the absence of the antibiotic that is present. If we put a specific concentration in there, we can determine if the bacterium is resistant or sensitive to one of the antibiotics. We can actually see that by the circles that we observe. If the circle is totally closed, it means the bacterium is actually resistant because it can grow in the presence of the antibiotics. If it cannot grow in the present, we have a sensitive bacterium. Now, I've talked a lot about diagnostics. These are the three major diagnostics that are executed nowadays. Let's go to our pig. In pigs, we actually have a quite complex story. It's not only in pigs, because we also have it in cattle. there's actually not one pathogen that is infecting the pig or the cow, but it's a mixture of viruses and bacteria. Of course, if we want to target all of these in one test, it becomes very difficult because for the PCR, we do require the sequence information, the genetic information. We need to be able to grow them for the bacteria. So there's actually quite some exclamation marks over here. And these are actually representing some problems nowadays because we can't just diagnose them all in one single test. We need to perform multiple tests to get this entire picture resolved in one diagnostic test. That's why we developed, and actually Pathosense was founded in 2020, a spin-off company of the university, in which we want to use the sequencing, in this case the nanopore sequencing, to actually look at the total picture. Not only target one specific pathogen, but really look at all viruses, all bacteria, and also the difficult ones, the ones that we can't grow, the mycoplasmas. In the future, and that's why these circles are in green, we actually also want to include the antibiotic resistant testing, because it's not there to date. The red circles, we are already able to identify all the viruses and bacteria within a sample. But the green circles, that's actually what I'm going to talk about in my talk today. Now, if I talk about genomics, of course, you need to think about, you need to know what is meant with genomics. If we see genomics itself, it means we're going to look at this genetic, if you remember the 4-letter code that I've been talking to you before, we really want to dig into this letter code. And we want to know which specific letter code is associated with a virus, with a bacterium, or with any organism we want to study. If we focus on one specific organism, it can be the mycoplasma, it can be a coronavirus, it can be anything, then we actually do genomics, because we want to target the information that is present within one specific organism. If we think about metagenomics, we actually are going to take a step back, because we're going to extract all the genetic material that is present within our sample. If you think, if for your self-test you have to take the nasal swab yourself, we would just get all the genetic material from this swab and use it in our further processing. That's what we call metagenomics. We're going to look at the bigger picture of all genetic material or viruses and bacteria in one specific assay. Now, the last part of my introduction, because I can't go to the analysis without introducing the technology I'm going to use, it's actually the Oxford Nanopore sequencing platform, which is a 3rd-generation sequencing platform. Now, I can tell you this device allows me to sequence. That's the only thing you need. Well, I'm lying because you also need a little cartridge inside. So you also need this one to do the sequencing. The nice thing is that, and they are indicated in green, I'm skipping the red one, it's in real time. We have long reads, so it's actually limited and unlimited. And this little guy here, and for many people, €1,000 will be a lot of money, but in science, it's not that much of money to actually start sequencing with only €1,000. Now, to go back to the red one, and people always ask me, like, what about the accuracy of Nanopore? Well, as you can see, we went from 75% accuracy, which means 25%, so 25 errors in 100 nucleotides, to 99% this year. So in a very short time, we actually improved in a very significant way. Now, I could explain this figure, but it's much easier to explain you with a little movie. Then you understand what the advantages are of this technology. As you can see, the same device, the same flow cell as I'm showing you. The DNA is indicated in blue. In pink, we have a so-called motor protein. Motor protein, what does it mean? It will help us to put the DNA through the nanopore itself. It will go onto the flow cell. We have a lot of these nanopores, 2040 and 8 to be specific. It will allow to bind to these nanopores, and there is actually the magic going to happen. I'm saying magic because if you start sequencing, it's really magic. It gets never bored. So it will attach, the sequence will go through, and there are also ions going through. So this means that there's actually a difference between the upper side and the lower side of the nanopore. You can see the ions flowing, And this disruption of the ions will allow us to measure a so-called current, the current that can be changed to the voltage. Here you can see the voltage. It goes at 400 base pairs per second. And exactly this sequence, actually this information that we obtain, can be used to obtain the ACTG code, because every of these 4 letter codes has its own specific size, and this allows us to actually get the information from the nanopore itself. Now, let's go to the objectives of this PhD. So the aim of my PhD was to actually look how can we use this metagenomics with nanopore sequencing, and what is the impact, its added value, but also the future needs. This was divided in 4 subparts, where we have the added value in respiratory disease complexes. If you remember the cow and the pig I showed you, it was quite a difficult story. Then we have the advantages and future needs in viral genomics, where we really dig into the needs for some viruses, because it's not always such a good story that we have. The third part is the genomic markers in non-fastidious bacteria. What is non-fastidious bacteria? The ones that are easily growing. So the ones that we can get the result actually in 24 hours. And then the last one, it's maybe the most difficult one, is getting the same information from difficult bacteria. So let's think about the mycoplasma that I talked about. It's very difficult to get information from those. So let's talk about science now. The first part I want to talk to you about is a study that we did together with colleagues in München, where we sampled 25 farms. So at these farms, we collected samples from 4 different age groups, from the suckling period to the end of the nursery period, and we took tracheobronchial swabs. This actually represents the lower respiratory tract, so it means we're not looking at the nose, but we're really looking at what is actually present within the lung system. Now, if you remember, there were some of them difficult to culture, there were some of them difficult to diagnose, and we're not looking at just one pathogen, but a mixture of different pathogens. So this was the results that we obtained for the viral part, in which you can very nicely see that we could see some age-dependent tendencies. So if you go from left to right, you see that some viruses, for example, the PRRS virus, PRRSV, that this one was actually going up quite high over time. It also becomes a quite important pathogen because it was also linked to the disease that we saw. They were coughing, they were sneezing. If we looked at the bacterial counterpart, because the nice thing is we are not stuck with only viruses, we can also immediately look at all the bacteria in the sample. So what do we get? We see that some bacteria seem to go down over time, and some bacteria seem to go up. The first one that we have is Glycerella, for example, seems to go down by the end of the nursery. This might be that maybe it's a good guy in the suckling piglets. We have no idea, because suckling piglets, they seem to be very healthy. They don't show coughing, they don't show disease. So what is this story? But we get a big picture. We get the all-in-one. Now, let me show you how the results would look like if we look at classical diagnostics. A lot is gone. So it's not complete at all. Well, I'm lying. This would be the result that you get from the traditional classical diagnostics. Pluses, minuses, it's positive or it's negative. So if there would have been a quantitative PCR, if you remember the dark black boxes that I showed you, we could determine if somebody had a low load or a high load. We can do that for specific viruses and bacteria for animals as well. Now, one of the disadvantages here is that I would have to pay for every single test. So every PCR that has to be performed has to be paid for. And on top of that, if we want to have a bacterial culture, we also need to pay for the bacterial cultures, the identification, and if we want to do, en plus, still some antibiotic resistance testing, we have to pay again. So at the end, this becomes a quite costly diagnostic if you really want to get the full picture diagnosed. Of course, you can choose as a veterinarian, I'm only going to look for one. Then you can be lucky or you can be not lucky if you detect something or not. So there are also some multiplex PCRs available. What does it mean? Where we can test everything in one go, but they are very limited to certain pathogens. They don't include everything. But in Belgium, for the porcine alternative, it's not available. For the bovine, we do have them available in Belgium, but for the porcine respiratory disease complex, it's not available. In Germany, it is, for example. Now, as you can see, there's an exclamation mark for this PRRS virus there. Why do I put it there? Because if you think about the human counterpart, if you get a flu vaccine, you get a live attenuated vaccine. That means the vaccine is alive, so it replicates, but it actually is no longer that virulent to yourself. The same is done for pigs, for PRRS, for example. So if you get a positive result, the question is if you actually detect the vaccine or if you're detecting a real ongoing infection. Because does this PRRS contribute to my coughing or sneezing, or is it just the vaccine that we're detecting? So let's go back to this original code, the 4-letter code that I talked about. The difference here is that the vaccine and the ongoing infection, they actually differ only in a few of these 4-letter code nucleotides. So they are not very different, but they are somehow different. That makes it quite difficult to target them in a PCR test, as I showed you before. Now, what do we usually have to do? Next to our classical diagnostics, the veterinarian has to request a sequence analysis in which we can compare if we have indeed a vaccination strain or we have an ongoing infection. In this example, they are very far away from each other. If you want to read this, the closer that the things are in the tree, because this is a phylogenetic tree, the more they are alike. The further they are away, the less they are alike. So the blue one is on top, the orange one is at the bottom, so it's very unlikely that we're talking about a vaccination strain here. Now, if it would be the other way around, the veterinarian would know this is my vaccine strain. So I actually don't have to treat for that because I'm just targeting my vaccine in my metagenomics, in my sequencing output. Now, if I put this next to all the other diagnostics that I showed you before, we get the time, which is approximately in the same range of the PCR. We do get the price in the range of a multiplex PCR. We can detect all Mycoplasmata C, but the sensitivity and specificity is slightly lower than the PCR test itself. We cannot yet detect the antibiotic-resistant testing. We can do the quantification because we get relative abundances, and we can also determine in a very, very accurate way if we have a vaccine or if we have no vaccine strain, because we get sometimes the complete viral genomic information, sometimes it's partial, but even with partial, you already can do a lot of analysis. If we continue to the viral genomics itself, I'm going to talk and dig a little bit deeper in some of the viruses itself. This is a picture of some of the pigs that we sometimes receive for diagnostics, and as you can see, there's some small mummified piglets on the bottom. They are actually fetuses, they are not piglets yet. And we know that in many of these cases, we actually have a porcine parvovirus type 1 infection. And why are they targeting this reproductive state? Because they do want to replicate in dividing cells that are in the S phase. So that's why if you have a pig, a young fetus, it's growing very, very actively, so there's a lot of the cells in the S phase. So that's why we have this parvovirus infecting the fetuses quite easily. Now, this means there's really a lot of the virus presence in these samples. That makes it quite easy to do the metagenomics because we have a very small genome, we have a DNA virus, and there's a huge amount of virus present, a very high load. So it means we can very easily detect and identify the genome and the complete genome of the virus. Now, this allows us to go and dig into the evolution of this virus. So by doing the metagenomics approach, or really looking at the genome of the parvovirus, we could estimate how the virus evolved over time. Now, what does this mean? We can see that there's specific changes on the virus itself. Think about the Omicron, think about the Delta. It's the same kind of story. We have specific mutations that are associated with, for example, escape of vaccination. Some people were more sick, some were, there were a lot of more transmissibility of the virus. This is kind of the same story. This virus is continuously evolving, but by having this genetic information, we can study it. We can really look deeper into the genome of this virus itself. And as you can see, we or nowadays having this blue virus, which seems to be nearly 100% abundant in Europe. We only have one specific variant circulating, but of course you want to have a good vaccine against that one, because if you can't vaccinate your pigs, you have a lot of problems, you have a lot of economic losses. So you might think, okay, how are we sure that this is true what we're looking at? Now, we can look at the pig populations, because more pigs means more hosts, means more piglets. So if we put this together, we could very nicely see that there was a very good correlation between the increase of the population of the virus, but also the increase of the pig population, which is indicated in yellow and red here. Now, why this steep decrease from like around 2000s onwards? Well, if we think about it, there's unfortunately not a lot of accurate data available for this. The vaccination degree and the health management really increased over time. So we know that more pigs got vaccinated, more pigs, actually we were more hygienic for the pigs and so on. So that actually nicely corresponds to what we see. Due to all the measures that the veterinarians and the farmers have to apply, we get a reduction in the virus itself. However, we do know that the blue one that I showed before, it's actually on the rise nowadays. Now, let's go to a more difficult story, because it's not always that easy to get this genomic data, because sometimes we have very dirty samples. And definitely in the case of swine influenza A virus, we often get the question, yes, but what's the subtype of this virus? So we really want to know which type of influenza virus is circulating in the pigs. And one of the sampling types that has been getting quite a lot of attention lately is the oral fluids. Now, you have to think these oral fluids, they are collected from a rope attached in the barn, and the pigs are just chewing on it. So there's not only pig material, but there's also fecal material, mucins, proteases, even food, antibiotics. So it's dirty. It's extremely dirty, the sample. So metagenomic sequencing, less efficient. We can't get the information that we want. However, there's a solution. We can target our virus that we're interested in. For example, here, we want to know the sequence of the influenza virus itself. Now, very lucky we are that the influenza virus helps us a little bit in here, because it actually has conserved sequences at the 3' and the 5' of its segments. So we could actually easily target them. If you think about knowing a sequence, we can target them with the molecular detection approaches, and then we can sequence them afterwards. So that's what we did. Important messages that we got from this study was that sample storage is extremely important. Don't store them at -20, because there's a lot of inhibitors in these samples. -80 is the way to go to. We also evaluated different protocols in which it seemed that the primer sequences are very important, but what might be more important is the ramping rate. What do I mean with ramping rate is, in a PCR, in the molecular detection, we go from a high temperature to a lower temperature, but of course you can go in a very quick way to this lower temperature, but you can also do it in a very, very, very slow way. And this is actually the ramping rate we're talking about. And this can have an influence on the primer binding to specific segments, but also to these secondary structures that are present. So it really helped us in getting better genomes. As you can see on the picture, the colored one is the improved protocol, including ramping times. The gray one is the one that we actually used without the ramping rates, so it is not that efficient in recovering the entire genomic sequence from the influenza. You might think, why do we want to know this information? It's actually quite important to know which influenza strains are circulating, because they are zoonotic pathogens. They can infect humans as well. So we want to know which strains are circulating in pigs. We actually want to know them for vaccination development, and it's very important from a One Health perspective. Now, that's why I changed this table a little bit, one little step back. It's a very good approach, but we have to think about the complexity of the sample, but also if it's a low biomass sample, we might have to consider to use a more targeted sequencing approach to get the information that we want. So we also have to think about the disadvantages of the technology. So let's go to the bacteria this time, and I will start with my question again. Who has taken antibiotics? I thought so, yeah. But let me ask the following questions. Question, who has taken antibiotics knowing against which bacteria? Oh, yeah, yeah, somebody in the back there, yeah. Some people, indeed, if you get to the hospital, a very heavy infection, you indeed know probably which infection you had, but many of you took antibiotics without even knowing which bacteria you had. Now, if I put these numbers on the screen, we have a quite big problem that is coming at us in the future. It's a huge burden that many people actually have infections with resistant bacteria, over 500,000, 33,000 of them die, and it costs us 1.1 billion euro each year. That means we have to spend a lot of money. So that's what we're paying for. So this problem is that there has been an increased antibiotic resistance amongst many different bacteria. And this has put a lot of pressure on the veterinarians, actually, because they have to reduce the use of many of the antibiotics. And that's why AMCRA, which is indicated here, has put specific guidelines in front, class A, class B, class C antibiotics. And class A means you can just use them. You don't need to do any diagnostic test. Class B means you can use them, but first you have to do the identification of the bacterium. Class C means you can use them, but you first have to do bacterial identification and tell us if it is a bacterium that is resistant or sensitive against a specific antibiotic. Now, of course, we first have to be able to do this, because as I said before, we cannot get this information from all bacteria that are actually circulating and that are pathogens. This is just to give you an idea of how many different antibiotics and how many different mechanisms that actually exist for bacteria to actually escape this use of the antibiotics. So there's a lot of different mechanisms that evolved to actually get them escaping the antibiotic treatment. Now, I will dig into the bacterial genomics, in which I first want to focus on the fact that we should collect proper data. If we don't have good genomics and we don't have good metadata available, it becomes extremely difficult to do this kind of analysis that I'm going to show you now. That's why we need to have good sampling in actually a representative region, here shown as Belgium, but we also need to have a proper technology to do the antibiotic resistant testing, for example. It can be the virulence that we want to assess. It can be anything of metadata that we want that is associated with disease, for example. Now, this can all be put together in softwares that are available. We can use databases, we can use de novo approaches, we can use all different types of softwares to actually put this data together, and then we get these kind of pictures, where we have, as you can see in the darker colors, we have a phenotype being resistance, virulence, and they can be very nicely associated with genes, genetic information that is present within the bacterial genome. Now, to put this in perspective of an example, a real-time example, we have the enterotoxigenic Escherichia coli samples that we tested. We know that there's increased resistance here, and what we did, we actually checked for a wide variety of antibiotics if they were resistant or if they were sensitive. Now, how do we do this? We can actually determine a non-wild type and a wild type infection, indicated in green and red. Green means they are actually sensitive, so we can use them, they will die. Non-wild type means they are actually showing resistance to the specific antibiotic. So as you can see, we have here the amoxicillin, in which we have a very big population that shows the non-wild type. In the cephalexin, for example, on the right, we have a very small population belonging to this resistant population. Now, we want to get this genetic information. We want to get rid of the culturing, so we want to know genetic markers to use in our metagenomics approach. So, as you can see, there's quite a nice collection of genes that we can use to show an association between genes and the phenotype. And indeed, actually the population that we see on the left, the non-wild type population, most of them harbor one of these genes that convert them resistant. Meaning, if we can target this gene in our metagenomics approach, we know that they will be resistant. The same accounts for cephalexin, in which we actually identified one specific mutation. It means one of these ACTG codes was changed within the 5 million of them, and this conferred resistance. So we have to go to a very accurate approach. In this case, it was only one mutation that conferred them resistant. Now, Important in diagnostics is to say how sensitive, how specific is your test that you're actually using. So we did these kind of calculations for all the antibiotics that we tested for, and we got to a nice 91% sensitivity, 95% specificity, but actually by moving our border a little bit more to the left, we got to an even better 94% specificity. So it might actually be that the population, that's the small number of bacteria just falling outside of the wild-type population, that they actually just belonged to this to the wild-type population and not to the non-wild-type population. If we would increase the number of samples here, we could resolve this problem and would definitely get a better answer there. Now, we have not done this for one antibiotic, but for a wide variety of antibiotics, not only in Escherichia coli, but also in Actinobacillus E. coli, which is one of the pathogens that is causing disease in horses. So this is actually applicable for all bacteria that are easy to grow, if we have a good data set and good metadata. The same can be done for virotyping. If we want to know if a specific virulence gene is present and associated properly, can we really say that it is a virulent strain or a non-virulent strain? As you can see, the darker color is the official golden standard that is used. It's the test that is nowadays used. It's a PCR. But on the other side, with the genomic information, we can get actually to this very nice association with the genetic information. The additional feature is that we could actually identify new features with our technology, not only sticking to the multiplex PCR that we have been using for tens of years now, but by using the genomics approach, we can also identify new information. It gives us more information than just sticking to this PCR tests. So let's go to the last part, which is a little bit the most difficult one, I would say, because we wanted to do the same thing for difficult to grow bacteria, which also have a difficult to sequence genome, to be honest. So let's dig into the genomes itself. Now, here you can see that building genomes of bacteria, it's actually putting a puzzle together. If you have a lot of pieces, let's say you have 1 million pieces, it's going to be very difficult to put the puzzle together. And imagine that there's hundreds of pieces that are looking exactly the same. So it becomes very difficult to put the puzzle together. Of course, if you have a puzzle of 1,000 pieces, it's going to be much easier to get this entire genome resolved, to get the assembly done. And that's exactly what we can do by using this long-read nanopore sequencing. Now, this is a puzzle, but this is a real-life example. That's how my genome looks like if I have only short pieces. And on the right, you can see the genome that was actually built from the very long pieces. A nice circular genome, how it should be. Now, one of the problems is that we had 10 errors with the short pieces, but we actually still had 1,000 errors, and if you think back my red remark that I skipped to the end, the accuracy of the long reads, it was 1,000 errors, so we were not happy. Because, as I just said, one mutation, one change can already confer resistance. So how can we be sure that these 1,000 errors will not cause resistance? So we trained a model, but it's a kind of complex situation, and I'm going to guide you through. This is how the standard situation is done. So if we have a normal bacterium, E. coli, for example, that's easy to sequence, we get our true sequence on the top, and we get our voltage difference represented like the squiggles. That's how they call them. If we base call them and convert these squiggles to the CACTG code, we get the true reference. We don't get any errors. Even if there's one mistake or one mutation indicated in purple, we know that the model will be able to very accurately get it out. If we would use this same model for, let's say, the Mycoplasma, which has a difficult to sequence genome, it would give us a lot of errors. Why? Because as you can see, there's this M indicated. It's small changes onto this ACTG code. And of course, the nanopore will not be able to detect the signal properly. So it will result in errors. Now, the nice thing with machine learning over time is that we can start to teach and actually make these programs learn how to process our data, learn them and teach them actually how the data should be looking like. And that's exactly what we did. We taught the program to recognize our specific difficult-to-sequence bacterial genome. So by training our own model, we went from sequencing errors to barely any. So we got to 10 errors as well. That's what we did, and so we could continue and identify specific point mutations in our mycoplasma genome that could be identified to be associated with antibiotic resistance again. Here it's very important because mycoplasma has no genes, no plasmids. In this case, we were in the bovine one, the Mycoplasmopsis bovis, and we actually know that they are all point mutation mediated. We also know that we need 2 mutations in order to actually get specific resistance, and if there was only 1 present, we saw that they were belonging to the wild-type population. Now, this is important information because if we would be treating one of these strains that is actually, or the infection that is on the left side of the graph, we would actually put additional pressure and we could actually get it to being a resistant strain in the future. So if we would only have done the phenotypic resistance check, we would not have known this information. So by doing this sequencing, we can identify potential strains that we would definitely not treat with a specific antibiotic. Now, the same story was told for Brachyspira. It's also a very difficult to sequence bacteria, and it is a causative agent of swine dysentery. So it also is a difficult bacterium because it also has a very strange buildup. It's very difficult to grow them because it takes really 14 days before you can really get a positive culture. Now, as I said, sensitivity specificity is very important, and also here we identified genes, but also point mutations that could help us to actually get to a genetic type of test and not relying on the phenotypic test, which would take us 14 to 28 days. So we actually had a quite accurate system that allowed us to detect the resistant strains in this Brachyspira hyodyssenteriae. Now, the interesting story there is that presence of a gene does not always mean that they are really resistant, because some mutations inside these genes might also mean that they are considered inactive. And that's exactly what we saw with this specific thiamulin and valnamulin resistant gene in Brachyspira hyodyssenteriae. So, as well, we didn't do this for only 1 antibiotic, but we did it for multiple, and these are actually all the results that we obtained for both Mycoplasmopsis and Brachyspira hyodyssenteriae. Now, let's go back to this graph, the overview that I gave you before. We can actually almost, I put it between brackets because we're not there yet. This is really the fundamentals that were set to do this genetic antibiotic resistant testing. We have the markers, we just need to imply them. I'm saying just, I'm looking where my colleague is who's going to take over, and she's sitting there, so she's going to implement them in the future. Good luck. So let's go to the conclusions. I just want to give you a general overview of what to think about in metagenomic sequencing. First, the sampling is very important. Sample choice, the veterinarian should really think about the proper sample, the proper diagnostic, and doing a proper differential diagnostics. Sampling device is also important. That's why we have our patented sampler. Transport is also super important. Store your samples the entire day next to you in the car at 21 degrees. Good luck, I can tell you there's going to be a lot of Pseudomonas in your sample. No, that's not how to do it. 4 degrees, always try to keep them at 4 degrees because it will prevent overgrowth of unwanted bacteria. Because I told you, if it's a complex sample, it becomes more difficult to get the information. Pre-enrichment is also important. Pre-enrichment, again, sampling device to get rid of specific material inside. Enzymatic removal. Why enzymatic removal? Because if we take a nasal swab, we actually also take our own genetic material, and we are not interested in our own material or the pigs or the cattle, so we have to remove it. And then the mucins, drugs, enzymes, all the inhibitors, we need to try to remove them, because otherwise we have some inhibition going on in our processing. The next step is the metagenomic sequencing itself. First, the at-random amplification should be really, really assessed properly, use the proper enzymes, use the proper protocols, and evaluate everything, validate everything. We can, if there's some troubles with some samples, think about the example that I gave you for the influenza from oral fluids, we can target them with a targeted enrichment protocol. This is optional, but it can help you to really unravel specific genomics information. And in veterinary medicine, we have to think in the cost-efficient way, because if it's too expensive, nobody is going to use it. So do we use nanopore sequencing? Do we use Illumina? Or do we use something else maybe in the future? We really have to evaluate and screen the potential in the field. Why am I saying this? Because if you think, look, now if you would request a multiplex PCR, you would actually pay the same amount of money for the sequencing, the metagenomic sequencing. So if you anyway pay for the multiplex PCR, I think it is actually a worth to pay for diagnostic test, because you get so much more. You get all viruses, all bacteria, and next to that, you even get the genomic sequences from the viruses. I do believe with the antibiotic resistance, with the virotyping in the future, that it's even going to be a better worth to pay for diagnostic tests. But as I said, my colleague is going to do and imply that as soon as possible. So another alternative, and that's why I put these next to me here, is that there are also other sequencing devices of nanopore. This one has 2048 nanopores. This one has less nanopores, because you can see it's a smaller one. This one can be reused. This is actually a one-time use flow cell. The disadvantages is that, one, its error is a little bit lower, but nanopore doesn't want to believe this. But we actually have the flongol, and it has a funny name, and the flongol has also lower pores, as I said. And this does not give us always the highest sensitivity and resolution, but I do believe that improvements in this type of flow cells will allow us to also get the cost dropping down. Now, one of the last parts is the bioinformatics. Host depletion is very important, because no enzymatic reaction is 100% efficient. There's always going to be residual material. Pathogen identification workflows, validating these workflows, it's extremely important, because all the databases are full with trash, and you want to get this trash out of it. Viral subtyping as well. Viral subtyping is also very important to consider, because people want to know this information. Think about the story I told about the PRRS genome. Is it a vaccine or is it a wild type infection? The influenza, we really want to know which subtype it is. So that's very valuable information. Reporting should be very easy to interpret, because if I give this zoo of bacteria and viruses as an output to the veterinarians, they will go crazy. So there should be a kind of curation, and we should assist them and support them in interpreting this data, because there's so much information coming out of it that we really have to scale it down. But of course, information might be very important from an academic perspective or company aspect. But for the veterinarian, we also need to make sure that they can use and apply the information that they get immediately in the field. That's why we have our own application that allows them to get all the data in an easy-to-interpret way. Then the last thing, it delivers a lot of solutions. Solutions in treatment. If we will have the antibiotic-resistant testing in the future, you will actually get, within 24, 48 hours, you will know which antibiotic to use. It will not matter if it's an easy-to-grow bacterium. Is it a difficult-to-grow bacterium? You will be able to get this information. There will, of course, still be a lot of R&D involved in this, but there are very, very good fundamentals to proceed with this in the future. We have the management, vaccination, the information of the viruses. It helps us to properly vaccinate the pigs, to vaccinate in time, to have this information. It really helps the veterinarians to actually dig into their diagnostics much deeper at a higher resolution. And the last thing is the monitoring and surveillance, because monitoring and surveillance is also very important. Like, I think we all would like to know if there's a new coronavirus coming in the future. So if we can monitor what is happening in animals, pigs, horses, in any animal, then it already will help us to understand what is happening. It might help us to really understand if there is some threat to human. It's really important from a One Health perspective. Now, I would say to infinity and beyond, an application for any pathogen, any host species, and any sample type. Thank you, and I'm now going to open for questions.